in the last lecture we were discussing about the chronic models of the inflammation, and the last model which we had seen, that is this one, sponge implantation technique. And this sponge implantation technique, the sponge with the help of cardboard of approximately 10 millimeter of diameter used to be cut, and they used to be soaked in the saline, sterilized in the hot saline, and for the induction of the inflammation, they are soaked in 1% carrageenan or 1% yeast, 1% zymogen, 6% dextron. So in this way, the sponge used to be then implanted in the animal. This diagram I think you might be remembering. ये लाइट बंद कर देगा. I think it might be more clear because further this light more. So the cork size which we used to prepare with the help of cardboard. So this is sponges approximately 10 millimeter of diameter, or as per our designing experiment, and that is why this type of pouch used to be prepared of about just double of the length, that is about of 20 mm of size, and up to 8 sponges can be kept, which are soaked in the carrageenan and other things which can be induce the inflammation. And two type of experiment can be performed, that is one, so here you can see the process, up to 8 sponges can be, so it depends upon the designing, can be implanted per rat. The dorsal incision is closed with the Michael clips or with the help of a suture, sterilized sutures. And for the short term experiment and long term experiment, for both the experiment this model could be utilized. So test drug has to be administered 1 day prior to the administration of the agent. So up to 3 weeks the chronic experiment can be performed, or that is called as long term experiment, or short term experiment can also be designed for this. So for the evaluation, for the estimation of the fluid phase of the sponge exudates, protein content, enzyme level, and biological mediators such as prostaglandin, as well as for the leukocyte migration, the sponges are removed already after 9 hours. So these many evaluating parameters could be utilized for this particular model. And for the studying the chronic phase of the inflammation, beside dry weight DNA indicating cell content, hexozyme indicating glycosaminoglycan content, and hydroxyproline indicating collagen content. So different content which we are determining in the chronic study having particular type of indication. So from this particular model, the short term as well as for the long term, when we can, in another way, that is modified method is also used, as I told, in a particular pouch up to 8 sponges can be kept. So out of 8, few are removed as an acute, and rest of the remain can be kept for a longer time period, that is for a period of 3 weeks for chronic study. And these are the different indication of finding. Then this was the thing which we were discussing about this arthritic model, adjoint arthritis. So arthritis is also a type of chronic inflammation, and when you will see the text, so there you will find that adjuvant arthritis in rat is considered to be an immunologically mediated and most frequently investigated model of chronic inflammation. And this adjuvant arthritis is a very closely similar to the clinical features which are associated in the human beings, that is of rheumatoid arthritis. So that is why this model is also chronic inflammation model, and most of the author also consider this model under the chronic inflammation. So the arthritis is induced, how we can induce? By subcutaneous injection of either Front's complete adjuvant, Front's complete adjuvant. What is this France complete adjuvant? Means two type of adjuvants are available, incomplete and complete. So here we are talking about the France complete adjuvant. So adjuvant is from Latin. The word adjuvant we know already well. Adjunctive anesthetic medication, you might be remembering of your UG period. So it came from Latin, which means help or aiding. So here with the adjuvant arthritis, it is aiding or helping in inducing the arthritis. So either we can use France complete adjuvant or we can use mycobacteria suspended in oil. So by both the way we can induce the arthritis in the experimental animal. The subplantar injection, as already it was demonstrated, once again you can see the picture of subplantar injection here. So subplantar injection of 50 to 100 microliter of this suspension. This suspension means France adjuvant, complete adjuvant or mycobacterium suspended in oil. So in the subplantar region of the paw, that is hind paw, 50 to 100 microliter of this suspension results in primary non-immune localized inflammatory response in the paw followed by secondary immune systemic disease. So initial phase and second phase. In acute phase it is non-immune, but in chronic phase it becomes immune systemic disease. So the local swelling begins in the injected paw within 24 hours. So this swelling is because of non-immune acute swelling. Reaches a peak on the day 4 or 5. So peak swelling used to be that peak swelling on the day of 4th or 5th, indicating the chronic inflammation. And that is why we consider this model after a week and become stabilized on the day 6 to 11. So the systemic disease usually start on day 7, is characterized by the swelling of the contralateral non-injected limb. Contralateral means opposite. If we are injecting in this one, then this is the contralateral. So non-injected limb से compare करते हैं and we just declare that there is a inflammation or not. Foot thickness and body weight changes can be monitored in the drug treatment group and compared with the untreated control. So that is we already know it very well. So loss in the body weight in arthritic condition has been reported to be associated with the reduced absorption of glucose and leucine in rat intestine. So this is the mechanism why there is a loss of weight. So that is because of decreased absorption of glucose and leucine in the rat intestine. This model allows evaluation of chronically administered drug against inflammation. So drug administration is done daily, starting a day earlier than injection of adjuvant. So the day when we give adjuvant, prior to that day, the testing material has to be injected for the evaluation, and we keep on continuing. So there is a delayed systemic response to the France adjuvant. What is the France adjuvant? That you have to find out, and from here you can take out ready-made a solution of antigen emulsified in mineral oil and used as an immunopotentiator, that is booster, or a suspension of desiccated mycobacterium in paraffin oil and mannite morolate that induces inflammation, tissue necrosis, etc. So you can find on the Google also what the France adjuvant, and this France adjuvant is nothing but the most concise way you may write like this. So which make this model superior to other model in assessing the efficacy of all type of potential anti-rheumatic drugs. So various modification which has been made in this particular method. First by Kai et al. 2006, where they have studied or investigated the role of various variables in the manipulation of induction of adjuvant arthritis in spongidolii rats and further characterize its arthritic feature. What they observed? They observed that the particle size and dose of Mycobacterium tuberculosis played a dominant role in the induction and severity of adjuvant-induced arthritis. So before 2006, the Mycobacterium suspended in the oil was not coded in this method. After 2006, this is the modification. Male rats manifested markedly more severe arthritic signs than the female rats. And that is why if somebody asks which rat or which sex has to be preferred in this particular model, then the male rats. So after subcutaneous inoculation with 500 microgram, MT means Mycobacterium tuberculosis, male rats develop pronounced arthritis with 100% incidence and low variable clinical signs. Even using 62.5 microgram Mycobacterium tuberculosis, adjuvant-induced arthritis was efficiently induced in male rats, and drug screening method characterized by up-regulated expression of interleukin 1 beta, interleukin 6 and TNF alpha. So we can also carry out the assay of these agents from after exfoliating or sacrificing the animal. As outburst spongidolii rats are much cheaper and more reliable, readily available than Lewis rats. So that is why they are commonly used. So SG rats means spongidolii rats and AIA means adjuvant-induced arthritis model. So it is an efficient and cost-effective arthritis model for studying the pathophysiology of arthritis in human as well as for screening novel anti-arthritic agent. In recent studies of Zheng et al. 2003, complete fronds adjuvant promoted the increase of IFN means interferon alpha and nitric oxide, gamma, and nitric oxide is in suppressing chronic and relapsing arthritis induced by persistence in rats, suggesting that adjuvant therapy may be a new strategy for the treatment of rheumatoid arthritis and other chronic inflammatory diseases. So here you can see with the help of the diagram. So this is the control and this is the situation of induced arthritis in the model. So it's a very effective model, and particularly in the male it is giving 100% result. And mechanism behind this model has already been disclosed. The involvement of a particular interleukin and interferon and other mediators are also established. Similarly, alternatively, another method could be used where neither if mycobacterium or getting the mycobacterium is not easy task. Similarly, fronds adjuvant complete adjuvant is also a little bit costly. So here another method could be utilized as an alternate. What is that? That is papaya latex induced arthritis. So the slow reacting anti-rheumatic drugs. Have you heard about it? Particularly the bhasmas, slow reacting anti-rheumatic drugs. Bhasmas, gold and other metals which are used as a slow reacting anti-rheumatic drugs, which are now part of the syllabus as per the new curriculum of the PCI in the undergraduate program also, such as gold, chloroquine, levetiracetam, penicillamine, etc. So these type of slow reacting anti-rheumatic drugs, they cannot be properly evaluated with the help of previous model. That would be very superior. So they have failed to show significant activity in the conventional experimental model. it has now been demonstrated that lysosomal enzymes play an important role in adjuvant-induced arthritis. So I hope all of you might be knowing the lysosomal enzymes, which are also present in the papaya latex. So these are the enzymes which are responsible. It is also mentioned here. These enzymes are known to cause tissue damage. Keeping in view the disease etiology, papaya latex, some composition, important compositions are mentioned of this papaya latex. The main composition is proteolytic enzyme, including the lysozymes. Many others are rolled over there. So, papaya latex induced model of experimental rheumatoid arthritis has been developed to test the anti-inflammatory activity of slow reacting anti-rheumatic drugs. 0.1 ml of 0.25% solution of papaya latex prepared in 0.05 molar sodium acetate buffer pH 4.5 containing 0.01% thymol. So when we see the say papaya latex, so papaya latex means this is the mixture which we have to prepare. And how to obtain the papaya latex? It could be obtained from the male flowers of the papaya tree, or it could also be obtained from the raw papaya fruit by technique of as we used to extract out the rubber. कट लगाएंगे तो हरा जो पपीता होता है कच्चा वाला उस पर हल्का सा कट लगाएंगे तो वो टपकने लगता है, उसको इकट्ठा कर लेते हैं, ड्राई कर लेते हैं, उसके बाद इस तरह से इसको बनाते हैं. So dry weight has to be mentioned. So what is papaya latex? So it is a prepared 0.1 ml of 0.25% solution of papaya latex. So what is 0.25? So जो solution है 0.25% वो कैसे prepare किया गया? By with the help of 0.05 molar sodium acetate buffer pH 4.5 containing 0.01 thymol. So it is injected into rat hind paw as already shown in the previous figure. The peak effect occurs at 3 hours and lasts for more than 5 hours. The method is sensitive for evaluating non-steroidal anti-inflammatory drugs like aspirin, ibuprofen and steroidal anti-inflammatory drugs, particularly SARDs, which do not show appreciable activity in adjuvant-induced arthritis and other model of inflammation, and that is why it becomes very important. Papaya latex-induced inflammation is known to be mediated through prostaglandins. Similarly, one more model is there by which we can induce the arthritis, that is zymosin-induced arthritis in mice. So in the early phase, intra-articular zymosin. What are zymosin? Zymosin particles are yeast cells walls of S. grevacea. Stimulate, so these zymosin which are obtained from the yeast cell wall stimulate edema formation accompanied by a massive neutrophil infiltration in synovial tissue and fluids of inflamed joints. In the late phase, this is in the early phase. In the late phase, chronic response is characterized by macrophages and lymphocyte accumulation. Articular inflammation is characterized by generation of tumor necrosis factor alpha, interleukin 1 beta, interleukin 1-8 or interleukin 8, interleukin B4 and prostaglandin E2 in the knee synovial space or extracts, clinically and in animal models. So Nido et al. In 2006 have induced zymosin-induced arthritis in mice by intra-articular injection of zymosin 500 microgram in 25 microerythropoietin saline into one knee joint under light ether anesthesia, and the contralateral knee injected with the same volume of beaker to serve as control. So how we used to proceed the investigation? The investigation has to be observed on the basis of zymosin intra-articular injection induced a significant increase in knee joint diameter within 6 hours, peaked within 24 hours and remained above control value for 20 days. Similarly, one more is there. All of you might be knowing about the Candida albicans. What is Candida albicans? Candida albicans. See. So most of the fungal infection is due to the Candida. So most common, most responsible fungi for the infection, systemic infection in the human being is Candida. So it could be introduced into the joint space as a result of the surgical procedure or during disseminated infection. Disseminated infection is a spread of localized infection. The infection which used to be localized. We always used to be remain in contact with such organisms, either bacteria, fungi or viruses. But due to our immune system, they used to be localized at one place. As soon as our immune system got weakened or threatened, they spread and cause a disease. So that is disseminated infection. So where it happens? In immunocompromised patients. So Yardonov et al. In 2004 have developed a mice model of septic arthritis. They have investigated the joint inflammation caused by inoculation of ICR-CD2. What is ICR-CD2? Of course, this is a specific type of variety of rat, a mouse, genetically variant mice of rat. So a mouse model. ICR-CD2 is nothing but a type of a mouse model. Inbreed mice pair with live Candida albicans. In intact mice and mice with collagen-induced arthritis. So they demonstrated that Candida albicans infection, when localized into the joint, cause rapidly progressive septic arthritis. This effect was associated with a strong swelling, a rapid influx of the polymorphonuclear cells, and elevated section of the cytokines, tumor necrosis factor alpha, interferon gamma by lymph node cells. The joint infection exacerbated the established collagen-induced arthritis, which correlated with an increased level of anti-collagen antibodies. This model could be provide an opportunity to investigate the crucial factor for initiating the disease as well as to find the therapeutic approaches. So, one more at last. Induction of accelerated arthritis by collagen or LPS. What is LPS? Lipopolysaccharides. So here which type of LPS we are talking about? Here we are talking about the bacterial LPS, bacterial lipopolysaccharides. So Chaudhary et al. In 2003 have induced and evaluated arthritis following the method of Cases et al. 1992. So chicken type 2 collagen. This is a collagen which we use to obtain from the chicken. Was solubilized overnight at 10 degrees centigrade in 0.05 normal acetic acid at a concentration of 1 milligram per ml and emulsified in Front's complete adjuvant at a ratio of 1:1. Chicken type 2 collagen with the help of this technique 1:1 in at Front's complete adjuvant. So using 0.1 ml of emulsion, each adult female specific type of this mice age 12 to 14 weeks weighing 20 to 26 gram was immunized by an intradermal injection at the base of the tail with 100 microgram of chicken type 2 collagen. 17 days later, the immune response to collagen was boosted by a subcutaneous injection in the back of the neck with 200 microgram of LPS, that is bacterial lipopolysaccharide, a strong stimulator of immune or inflammation. Solubilized in 0.2 ml PBS, phosphate base, phosphate buffer system. Clinical severity of inflammation, that is redness, swelling, joint deformities, was scored periodically on a scale of 0 to 3 in all 4 extremities. The maximum possible severity score was 12. The incidence and the frequency of arthritis was calculated as follows. So here this formula is given. So incidence was the number of mice having at least one affected paw divided by the number of mice per group. So number of affected paw frequency is equal to number of affected paws divided by total number of paws per group. So this is the formula by which we can calculate the score. The advantage of this method: number one, this model is specifically well suited for high throughput screening (HTS) to identify novel inhibitor of integrin VLA4. What is VLA4? The very late antigen 4. The end point of the model is reached within 21 days, whereas in other model it takes weeks to months. So comparatively faster. It limits the amount of test compound required for the study by administration of the test compound at appropriate time. These investigators administered antibody to VLA4 on day 16 to 22, the factor phase of the response. Therefore, the animal do not need to be treated from time to time follows collagen immunization. Administration of LPS causes a highly synchronized response with reduced variability. So this is the recent advancement. The efforts are continuously put to develop a new and safer and more effective anti-inflammatory drugs, and that is why the search to inhibit the inflammatory signal transduction pathway on mediators. So in present scenario, anti-inflammatory drug discovery is based on preliminary in vitro observation in a number of standard anti-inflammatory acids. Lot of restrictions are there nowadays to utilize animals. All these chronic models and several models what we have seen, they all give pain and suffering to the animal. And as per the rules of the CPCSCS instructions of OECD and all other things, the minimum utilization of the animal should be there, and that is why how we can minimize the use of the animals or such models. Before deciding a particular model, we may proceed towards the in vitro techniques. The success of the in vitro techniques can be the basis for utilization of animal models. So in present scenario, anti-inflammatory drug discovery is based on preliminary in vitro observation in a number of standard anti-inflammatory assays, in which the test compound produces unusually potent antagonism of inflammatory pathways such as arachidonic acid pathway and some cytokine cascades. The effective candidate drug in vitro test is later tested on whole animal. So as per this particular newer guidelines, first of all we have to study the in vitro methods, and successful result on in vitro assay method could be the basis for animal testing. So let us see some important in vitro anti-inflammatory methods. Inhibition of nitric oxide production induced by interferon gamma in mouse macrophages. So interferon gamma is a function, it functions as a primary activator of macrophages in addition to stimulating the natural killer cells and neutrophils. Interferon gamma has a role in activating anti-cancer immunity also. So what is the purpose and rationale? A test drug is screened to inhibit nitric oxide, which is one of the key mediator of inflammation. It is evaluated. Hydrocortisone, a well-known steroidal anti-inflammatory drug, is used as a positive control for such experiment, and potential candidates are compared against hydrocortisone for efficacy measurement. So standard or positive control is hydrocortisone. Procedure: inhibitory test compounds are dissolved in DMSO before addition to cell cultures. Final concentration of DMSO are capped at 0.1% or less than that. Controls with DMSO alone are also run alongside the test drugs. To obtain primary macrophages, female CD1 mice 5 to 10 weeks of age are injected intraperitoneally with 2 ml of 4% thioglycolate broth. 4 days after injection, peritoneal macrophages are harvested and processed. Cells are seeded in 96 well plate at 2 into 10 to the power 5 cells per well. उसको वाल बोल सकते हैं कुआं उसको, and incubated for 48 hours with 20 nanogram per ml interferon gamma in the absence of inhibitory test compounds. So this is a type of microbiological assay you might have been performed, the wall method. So measurement of nitric oxide production in mouse macrophages. So nitrite accumulation used as an indicator of nitric oxide production in the medium and is assayed by the Grice's reaction, in which this reagent 100 microliter is added to 100 microliter of each supernatant from interferon or inhibitory test compound treated cell in triplicate. The protein determination is performed by Bedford protein assay, very well known to you. That is a colorimetric method with the absorbent maximum moving from 470 to 595 nanometer. The plates are weighed at 550 nanometer against a standard curve of sodium nitrate. Mast cell degranulation. For collection and isolation of mast cells, heparinized thyroid solution is injected into the peritoneal cavity of Sprague Dawley rat, peritoneal cavity. After abdominal massage, the cells in the peritoneal fluid are harvested and then separated through 38% bovine serum albumin. Cells are washed and suspended in thyroid solution with 0.1% BSA at a 1 to 15 into 10 to the power 6 cells per ml. The cell suspension is pre-incubated with test drugs at 37 degrees centigrade for 3 minutes. 15 minutes after the addition of compounds 48 by 80. Compound 48 by 80 is a mixed polymer of P-methoxy and methyl phenyl ethylamine cross-linked by formaldehyde, widely used for non-immunoglobulin E dependent stimulation of mast cells. So this is the ready-made compound available, 48 by 80, or standard compound for mast cells regranulation. It is also called as. So glucuronides 1 millimole. Phenolphthalein D-glucuronide in 0.1 mole acetic acid buffer, pH 4.5 is used as a substrate absorbance at 550 nanometer after alkalization. And histamine 0.2% O calidyl condensation in pH 12.5. Fluorescence is monitored at 350 to 450 nanometer after acidification. So in the supernatant, so in supernatant we can determine not only the mast cells but also the glucuronidase as well as the histamine. This is the method how we can determine at what particular wavelength. The total control is measured after treatment of the cell suspension with Triton X100. The percentage release determined is the index of anti-inflammatory activity. So how we used to harvest the cell. So this is the method which I found from a research paper. So heparinized carrot solution, सिरिंज में लिया, पेरिटोनियल कैविटी में उसको इंजेक्ट किया, इंजेक्ट करने के बाद वैल्यू उसकी फूल जाएगी, हल्के से मसाज दिया ताकि मिक्सिंग हो जाए और जो मैक्रोफेजेस हैं वो क्या हो एक्सट्रैक्ट होके सॉल्यूशन में आ जाए. फिर उसके बाद एनिमल को थोड़ा सा किल्ड करते हैं. किल्ड करने से क्या होता है कि उसके ऑर्गन्स जो अंदर के हैं पेरिटोनियल कैविटी के एक साइड हो जाते हैं और जो सॉल्यूशन है जो वो ऊपर आ जाता है और फिर उससे सक कर लेते हैं. सक करने पे क्या होता है डैमेज भी नहीं होता ऑर्गन. और ये इस तरह से डेमोन्स्ट्रेशन के पॉइंट ऑफ व्यू से बिकॉज इन दिस पर्टिकुलर स्टडी द एनिमल वाज sacrificed. So here you can see the ballooning of peritoneal cavity. पहला A syringe filled with the STS, that is heparinized carrot solution. B to E, the injection technique you are seeing, and F, this process results into the balloon peritoneal cavity. This is the method I told you. In this way, without damaging the internal organ, we can extract out the fluid. So this is the diagram which is showing the extraction of peritoneal cell derived mast cells. The mouse is carefully shaken into the palm of the operator. The air is removed with a syringe. So this is the method of harvesting. In this way it should be harvested. Next is adhesion assay. So the vesicular protein VCAM, that is vascular cell adhesion molecule 1, ICAM1 intracellular adhesion molecule. So vesicular cell adhesion molecule VCAM and ICM intracellular adhesion molecules. So they are the endothelial cell adhesion molecules of immunoglobulin G superfamily. And E-selectin, which mediate adhesion, are investigated in primary cells derived from umbilical vein, and a microvascular cell line HMEC1, owing to their prominent role in the regulating leukocyte extravasation. So adhesion assay using cultured HUVECs, that is umbilical vein, or human dermal microvascular endothelial cells are the best approximately for the situation in vivo. लगभग इसका जो evaluation है वो in vivo के बराबर ही माना जाता है. So cell-based ELISA assay for inducible adhesion molecule in human endothelial cells have been developed. So what is the procedure? For assessment of endothelial cell neutrophil adhesion, TNF alpha stimulated HUVC and SDMEC are incubated with test drug at concentration from 10 to the power 6 to 10 to the power 5 mole for 20 minutes. It's not neutrophils. So they are added and allowed to adhere for 20 minutes. Adherent cells are lysed by 0.5% CTAB, that is cetyl trimethyl ammonium bromide, and fluorescence measured at 485 nanometer in an ELISA reader, which is a quantitative measure of the activity present in the test drug. Then platelet neutrophil adhesion, that could be achieved through thrombin activated human platelets are incubated with the drug at 20 degrees centigrade for 10 minutes and mixed with neutrophils at a ratio of 10:1. Neutrophils with 2 or more number and 1 or no adherent platelets are counted as the index of activity. Test drug block the adhesion with respect to control. Neutrophil adhesion to hypoxia stimulated procaine AOT. So procaine peak. So procaine AOT are stimulated by placing them into PBS gassed with 9,2 nitrogen and fixed between a Teflon block and a stainless steel plate with the drilled holes. Neutrophils and the test drug is added onto the luminal site for 90 minutes. Adhesion is blocked by the test drug and the adherent cells are lysed to assay the MPO activity with the help of a specific wavelength. So LPS, as we had seen earlier, induce expression of INOS protein, nitric oxide production, TNF alpha expression, and MAP kinase in mouse macrophages. So this is the modified new method given by the Lee et al. 2007, which has successfully cultured the murine macrophages raw 264.7 and cells at 37 degrees centigrade in this particular DMEM containing 10% fetal bovine serum. Penicillin 100 units per ml and streptomycin sulfate 100 microgram per ml in a humidified atmosphere at 5% carbon dioxide. So cells are treated accordingly and expression of INOS protein, NO production and TNF alpha determined by Western blot experiments. The expression of P38 MAP kinase NFK beta was also observed at the same dose of LPS by Western blot experiment. This is oxygen superoxide radical generation by PMNS, polymorphonuclear cells. So generation of excessive reactive oxygen species has been implemented in a number of diseases including the inflammation. That is the concept behind it. So leukocyte is specifically the polymorphonuclear cells, monocytes and the vascular endothelial cells are rich source of reactive oxygen species generation, contributing to the development of several degenerative diseases including the arthritis. Therefore, an in vitro model of superoxide generation by polymorphonuclear cells has been used by Diagene et al. 2006 for screening the compounds for potential anti-inflammatory activity. These investigators have cultured the polymorphonuclear cells and challenged them with a concentration of 10 to the power minus 7 mole NFLP, N-formyl methanoyl leucyl phenylalanine to optimally generation of superoxide radicals. Superoxide production was determined spectrophotometrically by measuring the superoxide dismutase inhibitor. So that indicate the reduction of cytochrome C reduced. So that give you the idea about the generation of superoxide radical. So similarly, molecules of the adhesion molecules can also be estimated by this particular method. So polymorphic, this is the procedure. Polymorphonuclear cell suspension are prepared from peritoneal exudates obtained 17 hours after intraprotein injection of 10 ml 6% sodium caseinate into sphingidylate rats. The cells are washed twice in a gauge balanced salt solution and resuspended to a final concentration of 10 into 10 to the power 6 cells per ml. The test compounds and the standard are dissolved in GBPS, formyl L-methanoyl and leucine, that is FMLP, is dissolved in DMSO. The further dilution are made up to final concentration of 10 to the power 7 molecule molar FMLP in GBPS. Before addition of FMLP, the cell suspension are pre-incubated for 10 minutes with the drug. So results are expressed as a change in the transmittance measured in millimeter or recorder. The mean peak untreated cells is set 100%. COX assay, this is cyclooxygenase, we know very important enzyme in the arachidonic acid metabolic pathway. So it exists in two forms, cyclooxygenase 1 and cyclooxygenase 2, which are constitutive and inducible in nature. So constitutive form is associated with physiological function, whereas inducible form plays a pathological role. That is housekeeping function, the prostaglandins are involved, and pathological role, particularly COX2 play important role. The in vitro assay for these enzymes may be carried out as under COX1 assay. The assay is performed by the method of Redel et al. 1994. 10 microliter of sample solution is added to 190 microliter of 0.1 molar of L-adenylyl dihydrogentratate in 10 micromole of hematine. After adding 0.2 units of COX1, it is pre-incubated for 5 minutes and 5 micromole of arachidonic acid is added. The incubation at 37 degrees centigrade is stopped after 20 minutes by adding 10 microliter of 10% formic acid. So here the nutshell, first of all, by putting the COX1, the arachidonic acid, we allow for a particular time period to remain in contact COX1 with the arachidonic acid so that it can induce a certain amount of prostaglandins within particular time period. As soon as you add 10 microliter of 10% formic acid, the reaction stops. The prostaglandin E2 concentration is measured with prostaglandin E2 enzyme immunoassay. COX2 assay, drug can be tested for COX2 inhibitory activity spectrophotometrically by measuring the velocity of oxidation of NNN, that is tetramethyl p-phenyl adenamine. So that is abbreviated as TMPD. So TMPD is oxidized during the production of prostaglandin G2 to prostaglandin H2. So the assay mixture consists of 100 millimole sodium phosphate, 1 micromole of hematine, 1 milligram per ml gelatin, 2 to 5 microgram per ml purified COX2 and 4 microliter of test compound in DMSO. So total volume of assay mixture is 180 microliter. This is then pre-incubated for 15 minutes at 22 degrees centigrade and then 20 microliter of a solution of 1 millimole arachidonic acid and 1 millimole TMPD in the assay buffer is added. The assay buffer contains the assay solution except hematine and enzyme. The absorbance at 610 nanometer is measured over the first 36 seconds and percentage inhibition is calculated. The non-enzymatic oxidation of TMPD in the absence of COX2 is also observed and subtracted from the value obtained. So this is the COX-1 and COX-2 assay. Cell-based receptor gene assay, that is also type of biotechnological technique. Chinese scientists Zhou et al. 2006 have established a cell-based drug screening system based on transcriptional regulation of microsomal prostaglandin E2 synthetase 1. COX-1 and COX-2 for discovering selective down regulation of MPEGES1, the upstream regulatory sequence of MPGES, COX-1 and COX-2 was respectively cloned into PGN3B new vector containing luciferase gene and new mycin resistance gene. After that, the recombinant receptor gene vector named this were respectively transferred into A549 cells, and stable cell lines M1, M2 and M3 were obtained. They detect, then there is a detection of samples by testing luciferase activity of M1, M2 and M3 cells in microliter vials to identify compounds that can selectively downregulate the MPGES1 expression. The compound which had more than 40% inhibition ratio of M1 and less than 20% inhibition ratio of M2 and M3 cells could be regarded as hits. This in vitro cell-based receptor gene assay is useful in screening selective downregulation of MPGES1 expression. So this is cell-based receptor gene assay. TNF alpha antagonist, tumor necrosis factor alpha. So that drug that can antagonize the activity of TNF alpha as a mediator of inflammation could be the of clinical value in combating the fatal effects. So what is the procedure? So 24 hours before the initiation of the experiment, L929 cells are harvested. These are cell line studies. L929 cells are harvested from stock cultures and are plated in 96 vial culture plates and incubated at 37 degrees centigrade and 5% carbon dioxide in air. Cells are then pre-incubated for 30 minutes with test substance or solvent before TNF alpha is added. After an additional incubation time of 20 hours, the culture plates are flicked out and the remaining living cells are lysed. After 30 minutes incubation at room temperature, 100 microliter of LDH reagent gives to each culture vial. After 15 minutes, enzyme activity is determined. Photometrically at 490 nanometer. So these are the in vitro techniques where TNF and alpha antagonist, cell-based receptor gene assay, cyclooxygenase assay, COX-1 and COX-2, FMLP induced adhesion of polymorphonucleolymphocytes, superoxide radical by production of oxide radical by polymorphonuclear cells, which used to be induced by FLP. LPS induced expression of INOS protein, NO production, TNF alpha expression in a specific mouse kinase, mouse macrophages, and different adhesion molecules, that is called as adhesion assays. Macrophages, measurement of nitric oxide production in mouse macrophages, and mast cells degranulation, and how these macrophages and other things used to be isolated, or so that is the technique. So this is the in vitro different techniques, and most of them are biotechnically oriented. Now let's start with the analgesics. So analgesia means pain. Analgesia, the agent which can reduce the pain. So the pain, it is unpleasant sensory and emotional experience. It is unpleasant to all of us, but emotionally experienced at a different level by different person of the same intensity of the pain. So that is a very complex pathophysiology of this pain, and to get the idea, like we had the idea of the inflammation is very essential, so that the involvement of the different component could be evaluated. So severe, basically if we see what is the pain, like inflammation, it is also a protective response of the body. So inflammation and pain, wherever the inflammation is there, the pain will also be there, because some of the mediators are same, and the mediator of the inflammations are also responsible or capable to induce the nociceptors, and that is why wherever the inflammation is there, the pain also used to be originate. So because it causes awareness of actual or potential tissue damage, and that is why it is one of the fundamentally protective response like inflammation. So how it used to be produced? It used to be produced by the excitation of the particular receptors. I already told you, the nociceptors. There are various receptors, but particularly when a specific receptor or any receptor which start giving rise the action potential, which gives sensation of pain in the central nervous system, could be termed as a nociceptors. So pain is produced by excitation of nociceptors or of their afferent fibers. These remarkable cells respond to a broad spectrum of physical, chemical stimuli. So the physical stimuli, heat, cold and pressure, which is also utilized for screening of the various, or development for the screening of various models of animal towards the analgesic activities. Chemical noxious stimuli, some of the chemical noxious stimuli which we can administer externally to induce the pain could also be the basis for the development of the models. In general, perception of the noxious stimuli is termed as nociception. So nociception is not exactly same as pain. Pain is subjective experience, and nociception is the incident which is taking place inside the body. It is carrying the pain impulses. Sometimes it depends upon person to person. Some may not be reactivity against that nociception, some are very much reactive. So this is the borderline difference between the nociception and pain. So nociception is not exactly same as pain. Pain is subjective experience and including a strong affective component, AC. It includes a strong affective component. That is psychological response toward the pain conveyed from the CNS to dorsal horn by descending pathway. I will show you later the ascending pathway and descending pathway. If you people are already having the idea of the gate, which is the most appreciable and accepted theory of the pain, then you can understand ascending pathway from periphery to the central nervous system, and central nervous systems many times it suppress the pain. I will try to explain later. So whereas nociception lacks the affective component. So this affective component is associated with the pain which is not there with the nociception. So nociceptors are bare or free nerve endings. They do not adopt or stop responding. To sustain or repeated stimulation, thus beneficial in keeping the individual aware of the damaging situations as long as it persists. Nociceptors are widely distributed in the skin, dental pulp, peristeum, joints, meninges, and some internal organs. वो एक अलग बात है, कंटिन्यूसली हम जैसे दर्द में जीना सीख जाते हैं और उसको क्या करते हैं, हमारा ब्रेन को हम ऑर्डर देते हैं कि अभी आप बिजी हैं, कोई पेन हो रहा होगा तो आप उसको थोड़ी देर सप्रेस करके रख सकते हैं. लेकिन नॉसिसेप्शन बंद हो गया, इट्स नॉट लाइक दिस. दे आर कंटिन्यूस सेंडिंग द सिग्नल. जो डिसेंडिंग पाथवे है, जो सेंट्रल कंट्रोल है, उसके द्वारा आप क्या कर सकते हैं, उसको महसूस करना बंद कर सकते हैं. गेट पाथवे भी उसके बारे में बात करेंगे. 3 मेजर क्लासेस ऑफ नॉसिसेप्टर्स: thermal nociceptor, mechanical nociceptor, and polymodal nociceptors. These are 3 major. And 2 type of afferent neurons which are associated with the nociceptor: A delta fiber and C fibers. Might be here many times. A delta fiber are small myelinated fibers that transmit impulses at the rate of 5 to 10 30 meter per second and associated with the thermal and mechanical nociceptors responsible for fast type of pain. Saltatory movement might be studied by you. There are 2 type of neurons existing in our body: myelinated and unmyelinated. In myelinated neurons, the action potential used to be propagated by jumping action. कूद के जाता है, इसलिए तेजी से पहुंच जाता है. जैसे धरती गर्म हो तो आपको वहां से यहाँ पहुंचना हो तो गम पर पैर मार के जल्दी पहुंच जाएंगे. ठंडी तो आराम से चल के जाएंगे. So this is similarly A delta fiber and C fibers which are associated with the nociceptors. So A delta fiber are myelinated fiber, that is why saltatory type of movement is there, and they are responsible for fast type of pain. C fibers are small unmyelinated fiber that transmit impulse at the rate 1 meter per second, that is range of 0.5 to 2 meter per second, and associated with the polymodal nociceptors, the 3rd type of nociceptors. They cause slow type of pain. So here you can understand 2 type of pain: fast pain and slow pain. For fast pain, myelinated A delta fibers are responsible, while for slow pain, C fibers, unmyelinated, are responsible. So let us see with the help of the table the characteristic of the fast and slow pain. So of course fast pain occurs at a first, immediately. Slow pain occurs second, but persists long time. Fast pain, sharp pricking sensation. कभी क्लास में से किसी ने चोटी काटी, खट से चिल्ला देते. That is an example of sharp pain. Slow pain, dull, aching, throbbing sensation. जैसे पेट में दर्द बहुत देर तक दबाए रहे, क्लास में बैठे उसके बाद बेहोश हो, है कि नहीं? तुरंत चिल्लाने का कभी मौका भी नहीं मिलता. Fibers, already told you, for fast pain, A delta fibers are responsible. For the slow pain, C fibers are responsible. Fast pain, thermal और mechanical. जल गए एकदम से चिल्लाते हैं, चूटी काटती, बताया, एग्जाम्पल दिया, that is. और slow pain, polymodal nociceptor. Some of the time पूछे पेट में दर्द हो रहा है, कहाँ हो रहा है, may not be able to locate. So that is a type of dull pain which is to be modulated by polymodal nociceptors. So that is why here the last stage you can see easily localized fast pain and slow pain poorly localized. Clear? Chemical mediators of pain are numerous, as we had seen in case of mediators of inflammation. But here some of the important mediators has been taken out because they are important. Amino acid glutamate. It is also acting as a neurotransmitter because it is acting as a neurotransmitter in both type of fibers, A delta fibers as well as the C fibers. So amino acid glutamate is the major neurotransmitter released by A delta fibers and C fibers. To elicit action potential. Without eliciting of the action potential, propagation will not take place, and until unless the propagation will not take place, nothing will reach to the CNS. No expression, no emotional, no reaction will take place. So that is why that initiation, we studied about the physiology of the muscle contraction, where acetylcholine begins that particular event. Similarly, here amino acid glutamate plays important role. Other mediators come from sources intrinsic to the neurons, including various neurotransmitters such as 5-hydroxytryptamine, substance P, which is only released from C fibers. They are not released from A delta fibers. A dull, throbbing type of pain, continuous or long-term pain, which is responsible substance P. And extrinsic to the nervous system, including substances from inflammatory immune cells and red blood cells, such as prostaglandins. And that is why pain is also associated with such events which are taking place in the body. Prostaglandins, kinins, cytokines. So cytokines के पहले जो काइनिन है वहां पर आना चाहिए था, ये थोड़ा मिस्टेक हो गया मुझसे. Bradykinin after this amino acid glutamate is more important in the physiology of the pain. Rest are indirectly associated, either these are prostaglandins or any other thing. Chemokines and ATP that are released following the injury to the tissue. So let us still talk about the bradykinin, then we'll discuss about this table. So bradykinin causes hyperalgesia, means where we are little sensitive become more sensitive. Same nociceptors become more sensitive. किसकी वजह से? Bradykinin is playing very important. It is making more sensitive. So by several mechanisms, 3 mechanisms I have taken to make you understand about this bradykinin role. First, it activates A delta and C fibers directly. So that is why the neurotransmitter release will take place from those fibers. Already discussed, that is substance P and glutamate. Second, along with the histamine, it contributes to inflammation. So further, inflammation persists for a little longer time. So immediate role will be followed by the role which persists for little longer time. So along with histamine, it contributes to the inflammation. The inflammatory mediators will continue stimulation of the pain. Third, it promotes the synthesis and release of prostaglandins from nearby cells. So these bradykinin and glutamate, that is why they are little bit more important with reference to the pain. Directly they are there. And here bradykinin not only having the direct action, but indirect actions are also involved through the second and third mechanism. Let us see with the help of this table endogenous chemicals activating or sensitizing the nociceptors. So potassium, which used to be released by the damaged cells, they activate the first order sensory neurons. First order sensory neurons I will show with the help of diagram also. पेरीफेरी से जो CNS मतलब पहला जो पार्ट कौन सा है, स्पाइनल कॉर्ड. तो ये है आपका फर्स्ट. स्पाइनल कॉर्ड से जो प्रोसेसिंग होती है वो सेकेंडरी, और उसके बाद जो ब्रेन है वो है टर्सरी. वहां पर बहुत सारा इन्वॉल्वमेंट. So that is the potassium which used to be released from the damaged cell, and first order sensory neuron used to be activated. Serotonin, that is 5-order system ऊपर जो हमने लिखा था, that usually the rich source or major source is platelets. So wherever the injury, प्लेटलेट का सबसे पहले टूटना, चोट लगा, घाव हुआ, प्लेटलेट्स टूटते हैं ताकि प्रोसेस ऑफ कोएगुलेशन और क्या बोलते हैं उसे, क्लॉटिंग में बिगिन. तो वहां से भी ये पेन सेंसेशन जो है वो स्टार्ट होता है. सो एंजाइम इन्वॉल्व इन द सिंथेसिस ट्रिप्टोफैन हाइड्रोक्सिलेज, सो इट एक्टिवेट आल्सो द फर्स्ट ऑर्डर सेंसरी न्यूरॉन्स. ब्रेडिकाइनिन, प्लाज्मा काइनोजन, and enzyme which are involved in the synthesis of the bradykinin is calcarein. So they also activate the bradykinin. The more detail has already been given to you. Histamine, all the living tissues, they have the mast cells, and degradation of the mast cells releases the histamine. More detail we had seen in the process of the inflammation. So pharmacological innervation of histamine, H1 receptor antagonist, diphenhydramine, chloride, Benadryl, etc. Good. Histamine mediated process could be relieved by them. Prostaglandins, we had already seen, arachidonic acid, COX-1, COX-2, that is cyclooxygenase involved. Sensitization of the sensory neurons used to be take place by the prostaglandins. And NSAIDs, if mainly the prostaglandins are involved in the pathogenesis, then prostaglandins could be prostaglandin inhibitors like non-steroidal anti-inflammatory could be a better choice for the treatment. Leukotrienes, which also used to be follow the arachidonic acid pathway due to the damage to the cells or the inflammation. LOX pathway is responsible for that, and they also sensitize the first order sensory neuron. Then substance P, the first order sensory neurons, and they sensitize directly the, so they are the neurotransmitter of the C values, as I told you. So this is the most nutshell what I can tell you. Then another classification already known to you, acute and chronic. So you can say pain can also be classified as acute or chronic. And on this basis we are having the models. The distinction between the acute and chronic pain is not based on its duration of sensation. Generally what we think, for a short time period it's acute, for a long time period it's chronic. But here it is not like this. That's why this is ill-defined basis of the classification of the pain, and you are finding it as a hidden in my presentation. But rather than the nature of the pain itself, acute pain which has its source soft tissue damage, infection and or inflammation will be short in duration. Chronic pain was originally defined as pain that lasts 6 months or longer. It is now defined as the disease of pain. The most common cause of the chronic pain include cancer, neuropathy and arthritis. Okay, so this is about the pain. Now let us see the mechanism, because the understanding of the mechanism may help you out to understand much more better way the different models. So nociception is the mechanism of transmission of noxious peripheral stimuli to central nervous system. नोसिसेप्शन क्या है? ये मेथड है कहां से? पेरीफेरी से सेंट्रल नर्वस सिस्टम को तक ट्रांसमिट करने का, नॉक्सियस जो स्टिमुलाई है उनको ट्रांसमिट करने का एक प्रोसेस है, मैकेनिज्म. So nociceptor fibers already explained, A delta fiber and C fibers terminate in the dorsal horn of spinal cord. Dorsal horn. Rather we can say spinal cord. All of you might have been little bit in your brain. It might be there. So superficial जो layers हैं, outer जो layers हैं, वहां पर terminate हो जाते हैं, deeper नहीं जाते. Dorsal horn of spinal cord. There they form the synapse with neurons, क्योंकि जहां पर एक न्यूरॉन एंड होता है फिर दूसरे को that is, so synapse with the neurons to the thalamus, that is known as the spinothalamic tract. यहां से अब thalamus brain के लिए जाएगा. Release of glutamate, already told to you, substance P, etc., contributing to the neurogenic inflammation. फिर ये एक सेकेंडरी सोर्स जनरेट होगा जिसकी हम बात कर चुके हैं. द थैलेमस ओनली कंट्रीब्यूट्स टू बेसिक पेन सेंसेशन और अवेयरनेस कि हाँ कुछ तो हो रहा है, कुछ गड़बड़ है. इट कैन नॉट डिटरमाइन द सोर्स ऑफ द पेनफुल स्टिमुलाई. कहां से आ रहा है, कौन भेज रहा है, नहीं. सिग्नल्स आर आल्सो सेंट टू द ब्रेन stems reticular formation via spino-reticular tract, and this spino-reticular tract जो बहुत दूसरे पार्ट्स में जो जाता है, सिग्नल्स पहुंचते हैं, वो मिलजुल के इस बात का डिसीजन लेते हैं कि आप उसको लोकलाइज कर पाएंगे या नहीं कर पाएंगे. ओके, सो दिस इज आल्सो अगेन इन नटशेल. ट्रांसमिशन इन द डॉर्सल हॉर्न, स्पाइनल कॉर्ड का डॉर्सल हॉर्न, वेंट्रल डॉर्सल दो हॉर्न होते हैं, इज सब्जेक्टेड टू वेरियस मॉड्यूलेशन कॉन्स्टिट्यूटिंग द गेट कंट्रोल थ्योरी. जो पेन की जो मोस्ट एक्सेप्टेबल थ्योरी है वो गेट थ्योरी है. गेट कंट्रोल थ्योरी सजेस्ट, व्हाट इट सजेस्ट, दैट द स्पाइनल कॉर्ड contains a neurological gate that either block pain signal or allow them to continue to the brain. उसको वहीं रोक देगा या ब्रेन तक जाने देगा. The gate in the spinal cord differentiate between fiber types when carrying the pain signals. Specifically, pain signals traveling via small nerve fibers are allowed to pass, while signals sent by large nerve fibers are blocked. कौन से स्मॉल है, कौन से लार्ज है? अब बता. So this is the gate mechanism. This is sorry to say, ये जो है वो कॉपीराइट इमेज है, लेकिन मैं खुद ड्रॉ नहीं कर पाया. So you are able to see the fast carried by A delta fibers and slow C fibers. तो सभी आप देख रहे हैं ये हो रहा है. और ये जो आप ये पेन गेट है, दिस इज द पेन गेट, किसी भी डायरेक्शन में ये जा सकता है. ठीक है, इट मे अलाउ, इट मे नॉट अलाउ. सो होता क्या है क्या ये गेट में? चोट सबको लगी, सबको लगती है ना. लगने के बाद सहला देने से, टच कर देने से गायब हो जाता है, हो जाता है. बच्चा रोया, मम्मी ने फूंक मार दी, पेन गायब हो गया. चोट लगी, सहला लिया, गिर गए, लग गई, सहला लिया, सही हो जाता है ना. That is the touch. Here you are able to see the leaves. Here this is the touch. जो सीधे कहाँ पहुंचता है ब्रेन में, और वहां से डिसेंडिंग पाथवे के थ्रू इनहिबिटरी सिग्नल्स भेजता है, और वो इनहिबिटरी सिग्नल्स अलाउ करते हैं या नहीं करते हैं, डिसीजन होता है ग्रे मैटर। ये है गेट थ्योरी, आइदर इट विल अलाउ और नॉट। सो ए टच may stimulate the ascending pathway, which in turn stimulate the inhibitory descending pathway, और जिन रिसेप्टर्स पे ओपाइड्स यानी नार्कोटिक एनाल्जेसिक जिन रिसेप्टर पैक करते हैं म्यूकोपाइन, तो वो सारे के सारे जो है दे आर मेंट फॉर endogenous substances in capillaries and all these things. They are naturally existing in our body. So natural analgesics are there, and they control mainly the reaction component. Well, that is why it was told to you that the pain mechanism is one of the complex mechanisms, and nociceptor, nociception, and the pain there is a. So this theory was introduced by Ronald Maslach and Patrick Wall in 1965. So descending inhibitory pathway from the midbrain and brain stem exert a strong inhibitory effect on dorsal horn transmission. Main transmitter in this pathway are encapsulin, 5-hydroxyhistamine, that is called as feel good neurotransmitter, and noradrenaline. Noradrenaline, fight or flight, responsible for fight or flight. So when you are fighting, other people are hurting, hitting, even then you continue, do not feel the pain that time. So a lot of complexities are involved. So this is involvement in that. So drugs in clinical use as analgesic belong to two main groups already known to you: narcotic or morphine group and NSAIDs group. Morphine-like drug produce analgesia by acting on the central nervous system, while analgesic antipyretic drug acting by both central as well as peripheral. But primary action is on peripheral, that is primary and secondary interface. One more pain pathway. Able to see it? So nociceptor, primary sensation of the primary receptors. They stimulate either A delta fiber or C fibers. So first order, as I told you, they reach to the spinal cord, the superficial layer. Then two things are taking place from there: the spinalothalamic tract, that is to the thalamus, which are allowed slow C fibers carrying impulses are allowed. They are not able to locate, but they will come to know that from somewhere the sensations are coming. With the help of limbic system and the hypothalamus, could be able to locate that somewhere where things are wrong, and accordingly your behavior behavioral and emotional responses used to elicit. The second tract is spino-reticular tract, so that increase the alertness, reticular formation. That is with the help of these three particular things, and The third order you are seeing, that is somatosensory cortex, localization and perception of intensity of pain. So there is a complex involvement of different parts of the brain. So the pain pathway, nociception transmission to several regions of the brain, including thalamus, reticular formation, hypothalamus, limbic system, somatosensory cortex, each of them carry out specific response to pain. So I think the next lecture we'll see the animals models of acute, chronic and in vitro methods. Already is 1.5 hours, more than that. Till here I think things might be clear.